Hello, this is my science fair video about my lab experience working on my project to determine whether brain matrix elasticity plays a role in glioblastoma cells' ability to resist chemotherapy. So up first, I had to synthesize the major component of my project, which is the biomimetic gel that sort of replicates mechanical components of brain tissues. So in this video, I am just following the protocol and including the three components of the gel, which is DTT, DMEM, and as well as the HA gel. After mixing all the substances that create the gel, using a pipette, I carefully placed the gel solution in a 12-well plate that will be used to seed cells onto later. And by using a cover slip, it helps make the gel stay in place while it stays in the incubator for 2 hours for the DTT reactions to complete. So after creating the gel, the next step is to seed the glioblastoma cells. I was given my own set of cells to work with for this project, and to seed them I first aspirated all of the old media and then added trypsin to remove cell adhesion from the existing plate. After adding trypsin, I put the plate into the incubator for 5 minutes. After 5 minutes passed, you can kind of see that the cells detached from the plate. So I added new media to prevent the excess trypsin from denaturing other necessary proteins in the cell. Next, I pipetted the solution into a vial tube to centrifuge. Centrifuging helps relocate all of the cells to the bottom of the tube, and it's important that I balance the centrifuge to ensure that I don't break the machine. So for this project, I centrifuged the vial for 5 minutes at 1000 revolutions per minute. So after centrifuging, you can see that all the cells have reached the bottom of the tube, and I actually think this is pretty cool. Now, next, I carefully aspirated the media plus trypsin from the tube, and I had to make sure that I did so without sucking up the cells during the process. So I kept the suction at a safe distance as well as tilting the vial. And once that was completed, I added new media into the tube. After putting the media into the tube, I mixed it thoroughly and excessively to ensure that all of the cells would disperse evenly. This is because later on I needed to count the cells to estimate the population that I had per milliliter of media so I could rationalize the proportions of cells that I would need to seed into the 12-well plate. To count the cells, I used a device known as a hemocytometer that is used to count cells in 4 regions to estimate the general population of the cell. In this clip, my mentor and I are calculating how many cells we have per milliliter of media, and we were going over what volume of media I would need to have to ensure that all 12 wells have at least 50,000 cells in each well.
Okay. So how do I calculate
it should be
how much I need?
500,000 divided by 960,000.
Okay.
Okay, so that should be...
So that's how many milliliters we need.
Yeah, to get it to microliters, I need to multiply it by 1,000.
Right, because that's the conversion.
So 520 microliters. So once I got the necessary volume of cell solution I needed to rationalize for the 12-well plate, I started to prepare the seeding process, first by pipetting the amount of fresh media into an empty vial, which we will later use to seed the cells. Then, I moved on to the vial with cells and mixed the cells in the media again to ensure that all of the cells are evenly dispersed. Then I pipetted the volume that we calculated to obtain the necessary amount of cells I needed for this overall trial and mixed it with the fresh media in the other vial tube to later seed the cells with. And then this last process is actually seeding the cells onto our 12-well plate with the gel, and once that was completed, we placed the cells in an incubator to rest overnight. So the next step is to actually apply the temozolomide drug treatment, but my mentor takes care of that step. So this clip is actually 1 day after treating the cells with temozolomide, and I'm currently applying live dead stain onto the cells in the well to use to look at the amount of live and dead cells there are using a fluorescence microscope. So this next clip is me collecting the data using a fluorescence microscope to take pictures of the amount of live and dead cells, which we will later use a program to count the cells to give us quantitative data. The same method was used to count the cell proliferation as well, and for migration, the microscope was capable of taking pictures of the cells in 15-minute increments to later be put together as a video that we can use to calculate the distances each cell moves. So that sums up my experiences in the lab and the way that I collected my data. Thank you for watching.